I'm Philip Barr. I'm the Executive Vice President for Discovery Research at InMed. I was a co-founder at BayMedica, where we set up BayMedica to capitalize on the technologies that we had to make cannabinoids, manufacture cannabinoids in yeast cells. My background is I got a PhD in organic chemistry in the UK, the University of Birmingham. And I did a postdoc here at UCSF in biochemistry, which is where I met the founders, some of the founders of the biotechnology industry. And I became eventually became the director of molecular biology at Chiron Corporation, which was one of the pioneers in the biotechnology industry, in that they were the first company to clone the HIV virus, human immunodeficiency virus, and also more recently we cloned the hepatitis C virus, and the principal investigator, Michael Houghton, received the Nobel Prize for that last year. And my role there, after joining as a chemist, was to since I was replaced by a DNA synthesis machine, was to manufacture diagnostic antigens, vaccine candidates, and therapeutic proteins in yeast, which I did for over a decade. And then following the Chiron years, I continued to work in the manufacturing of rare therapeutic proteins in yeast, and also in making small molecules, of which cannabinoids are in that family. So when I met with my co-founder chemist, and we talked about the fact that cannabinoids were a class of compounds that myself, Charles Marlowe, and also other people at the company had worked on extensively, it was pretty much a no-brainer that we decided to set up BayMedica, and we Chuck Marlowe had already had contacts with Shane, so it seemed like a really great threesome to set up this company to make cannabinoids, manufacture them in yeast, initially for both initially for the consumer products market, which we have we've successfully done, but also to bring in the synthetic biology process to lower the cost of goods of the main cannabinoids, but also to set up the systems that could make what are now really regarded as rare cannabinoids, which we can make at high purity, all of them, and manufacture them for the consumer market. Making cannabinoids in yeast was not as easy as we thought it was going to be, but we did crack the problem several years ago. And having cracked the problem, it became apparent to us that we could not only make large quantities of cannabinoids for the consumer markets, we had already contemplated the fact that we could make high purity cannabinoids for clinical use in humans and companion animals, perhaps. However, it became apparent that we could also make even more rare cannabinoids, known as analogs of cannabinoids, which we predicted then, and we have found since, to actually be highly active, and in some cases more active than the natural cannabinoids. So we're focusing on those two areas: the consumer products, where we can make multi-hundred kilograms of the products. We're probably the only company that can do that right now, and we do that through chemistry. And eventually, as we move these products into our yeast manufacturing systems, we will be able to continue to make them in the 100-kilogram range. And then we will also be able to make any analog that we would like to make for testing in the hundreds of human clinical indications that exist for treatment using cannabinoids.